So, dear students, good morning to everyone. We have started a metabolic study in medicinal chemistry 1 lecture. So, up till now we have covered phase 1 reaction, various type of oxidative conversions we have seen in that. If there is presence of aromatic ring, whether the aromatic ring is attached to electron withdrawing groups or electron donating group, how the carbonyl carbon atoms, that is aldehyde and ketone, they are converted into their corresponding alcohols. So these type of hydrolysis conversions, oxidative conversions, we have seen up till now in the case of medicinal chemistry 1, principles of metabolic reactions in that. Now we are going to see the second type of phase 1 reaction, that is reductive reactions. Up till now we have seen first and main important part of phase 1 reaction, that is oxidative conversions. Oxidative conversion of aromatic rings, oxidative conversion of amine rings, if primary amine, secondary amine, tertiary and quaternary amine, oxidative conversion of carbonyl compounds, oxidative conversion of if amide compounds is there. So these conversions we have seen how those if the drug molecule have these type of rings or structures in their compounds, so how they are going to metabolize in our body. Now we are going to discuss a second and very important type of metabolic reaction, that is reductive reaction. Now we all know the difference between oxidation and reduction. So what type of reductive reactions are proceeding in our body? So these reductive reactions are going to play very important role in the metabolism of many compounds. आतापर्यंत आपण बघितलं की ऑक्सिडेशनने जनरली जास्त प्रमाणात कंपाऊंड्स आपल्या बॉडीमध्ये मेटाबोलाइज होतात. नाउ वी आर गोइंग टू सी in spite of oxidation there are compounds or the molecules which are metabolized through reductive reactions also. Now what type of reductive conversions are taking place in our body? Reduction of carbonyl functional group. कार्बोनिल फंक्शनल ग्रुप मध्ये कोण येतं? तिघ जण येतात, अल्डिहाइड्स, कीटोन्स आणि ऍसिड्स, कार्बोक्सिलिक ऍसिड्स. सो या टाइपच्या फंक्शनल ग्रुप प्रेझेंट असणारे ड्रग मॉलिक्युल्स, they are going to metabolized by, we have seen them in the oxidative reactions also. Some carbonyl compounds are converted into acid derivatives, but some of the drug molecules in which there is carbonyl compounds are there, they are reduced, they are metabolized by reduction mechanism also. पण कशा पद्धतीनं? By bioreduction. Bioreduction means if enzymatic conversions असतील तर. कार्बोनिल कंपाऊंडचा प्रेझेन्स आपण ऑक्सिडेटिव्ह कन्व्हर्जन्स मध्ये पण घेतला, but it is chemical reduction, chemical conversion, chemical oxidation. But here if the drug molecules contain carbonyl compound आणि त्यांचं मेटाबॉलिझम जर एन्झायमॅटिक कन्व्हर्जन्सने होत असेल, बायो रिडक्शन होत असेल, सो दोज टाइप ऑफ रिएक्शंस आर कमिंग अंडर द कॅटेगरी ऑफ रिडक्टिव रिएक्शंस. नाउ व्हाट हॅपन? बायो रिडक्शन ऑफ कार्बोनिल कंपाउंड्स जनरेट अल्कोहोल डेरिव्हेटिव्ह. नाउ वी ऑल नो कार्बोनिल कंपाउंडचं रिडक्शन झालं की इट इज कन्वर्टेड इनटू एन अल्कोहोल डेरिव्हेटिव्हज. सो what type of reduction? Bioreduction. Bioreduction means presence of biological enzymes. ते reduction particularly कोण करतं? Biological enzymes करतात. So this is with respect to the carbonyl compound. If bioreduction of carbonyl compound is done, they are converted into the alcohol derivatives. If bioreduction of azo and nitro compounds, presence of nitrogen nitrogen triple bond, presence of nitrogen oxygen double bond, अशा पद्धतीचे स्ट्रक्चर्स असणारे ड्रग मॉलिक्युल्स, दे आर लीड टू दी अमाइन डेरिव्हेटिव्ह. सो दे मे बी कन्वर्टेड इनटू देअर करस्पॉन्डिंग आयदर प्रायमरी सेकंडरी अमाइन मध्ये त्यांचे कन्वर्जन्स केले जातात. दी हायड्रोक्सिल और अमाइनो मोइटीज ऑफ द मेटाबोलाइट, दे आर मच ससेप्टेबल टू दी कॉन्जुगेशन दॅन दी रिडक्शन किंवा ऑक्सिडेशन. सो जनरली सी आता काय बघितलं आपण बायो रिडक्शन कन्व्हर्जन्स करून किंवा कार्बोनिल कंपाऊंड नायट्रो आणि अझो कंपाऊंडचं कन्व्हर्जन करून those type of drug molecules are converted into their parent compounds किंवा simpler compounds. That's why these type of reactions generally go for what type of conversion? Drug elimination. That means reductive conversion किंवा reductive metabolism मुळे generally ड्रग मॉलिक्यूलचं कन्व्हर्जन अशा मॉलिक्यूल मध्ये केलं जातं की तो मॉलिक्यूल इझिली आपल्या बॉडी मधून एक्सक्रीट केला जाईल. सी फेज 1 ऑक्सिडेशन मध्ये व्हॉट वी हॅव सीन दॅट अ ड्रग मॉलिक्यूल इज कन्वर्टेड इनटू द ऍक्टिव्ह मेटाबोलाइट. दॅट्स व्हाय इट इज गोइंग टू गिव्ह इट्स फार्माकोलॉजिकल ऍक्शन. For what purpose we have taken that drug molecule? That type of conversion, majority of the case will be done by phase 1 oxidation. But here, in the case of reductive reaction, it will convert the drug molecule into drug elimination, or which will facilitate the drug elimination. Now, reductive reaction. What type of conversions will be done under the reductive reaction? So reductive reactions of aldehyde and ketone. So reductive reactions of aldehyde and ketone. The carbonyl moiety, reductive reaction of aldehyde. Aldehyde and ketone contain what type of functional group? Carbonyl functional group. So the reduction of carbonyl functional group, for example, aldehyde and ketone, they are undergo this type of reductive reaction in majority of the drug molecule. Including this, many drugs and their xenobiotics. Xenobiotics means active metabolite of that drug molecule undergoes oxidative deamination and produces their corresponding aldehyde and ketone. So these metabolites are undergo oxidation and some metabolites be eliminated through reductive reactions. When see already we have discussed just now, generally aldehyde and ketone, त्या aldehyde आणि ketone molecule मध्ये if they are going to involve the phase 1 conversions, ते कशामध्ये कन्वर्ट केल्या जातात? ऍक्टिव्ह मेटाबोलाइट मध्ये कन्वर्जन केले जातात. आणि त्याच अल्डिहाइड अँड कीटोन मॉलिक्यूल वरती जर रिडक्टिव्ह रिएक्शन प्रोसीड झाली, so it will get converted into an eliminated form of the drug molecule. That's why they are going to convert into an which will facilitate the elimination of the drug molecule. आणि ज्या अल्डिहाइड आणि कीटोन मॉलिक्यूल वरती दोन्ही रिएक्शन्स होत नाहीयेत, ऑक्सिडेटिव्ह फेज 1 रिएक्शन इज ऑल्सो नॉट देअर as well as ड्रग इलिमिनेशन किंवा फेज 1 रिएक्शन रिडक्टिव्ह रिएक्शन पण ज्या aldehyde and ketone molecules वरती proceed होत नाही, those type go further for the conjugation reaction. See, some aldehyde and ketone किंवा some carbonyl compound containing drug molecule go for phase 1 reaction. If phase 1 reaction will not be there, then they are going for reductive reaction in the presence of biological enzymes, bioreduction. And if both these cases are not there, then they are go further for the conjugation reaction. Then what happens when the aldehyde molecule is reduced? It is converted into an primary alcohol, and we all know if ketone molecule is reduced, it is converted into an secondary alcohol. So, and diverse soluble enzymes. See, already we have discussed reductive process कुणाच्या presence मध्ये होते आहे? It is done in the presence of biological enzyme. So कुठला enzyme actually take part करतं या processes मध्ये? So the name of the enzyme is aldoreductive is the enzyme which is responsible to carry out the bioreduction of aldehyde and ketone. So हा enzyme काय करतोय? It will convert the drug molecule into a form of the compound which will facilitate the elimination of the drug molecule. Now, reductive reactions. Reduction of aldehyde and ketone. So what happens if the reductive reactions are done on the aldehyde and ketone molecule? It will convert the aldehyde molecule into primary alcohol and the ketone molecule into the secondary alcohol. That's why they are converting the drug molecule containing aldehyde or ketone functional group into their corresponding metabolites, primary alcohol or secondary alcohols. That's why they will facilitate the elimination of the drug molecule. Elimination means what? They are easily excreted out of our body. Now, example of such compounds which are nowadays used for the treatment that are propranolol. What happens? When propranolol, as it is going to contain an amine functional group, see? So what happened? We all know propranolol molecule is first of all go for N-dealkylation. So after N-dealkylation, it is converted into an primary amine derivative, and the name of that primary amine derivative is disisopropyl propranolol. Now after that, this isopropyl propranolol will be converted into an aldehyde intermediate, and this aldehyde intermediate, if it is go for phase 1 oxidation, see दोघांमधला difference दिलाय. If it is go for phase 1 oxidation type of reaction, the propranolol aldehyde derivative, it is converted into naphthoxyacetic acid. If it is go for phase 1 oxidation, aldehyde functional group is converted into acidic functional group. म्हणजे oxidation reaction झालेली आहे. Which type of oxidation? Phase 1 oxidation containing aldehyde functional group or carbonyl compound. So they are converted into naphthoxyacetic acid. And the same drug molecule containing carbonyl functional group, if it is going for reductive reaction, it is converted into propranolol. But see here the dotted line is shown. That means this aldehyde molecule, majority of the case, it will go for phase 1 oxidation reaction, but to the minor extent, it will go for the bioreductive conversions to the minor extent and get converted into the propranolol glycol. Now, what is the category of the propranolol drug molecule? It is used as a beta blocker for the treatment of heart attack and other arteries and vein-related diseases, cardiovascular problems. Now, the same example containing aldehyde and functional group. Second example, acetohexamide. Now, it is an N-sulfonine urea derivative for the treatment of diabetes. So what happened for this acetohexamide? See, it will contain amide functional group containing carbonyl functional group. See, this is the structure. It is contain an ketone functional group. Now, generally, ketone functional group will convert them into the what type of it is converted into secondary alcohol. So see here, the ketone functional group is reduced by bioreductive enzymes and converted into their alcohol derivative, that is S-hydroxyhexamide. So this is the reductive conversions done on the acetohexamide drug molecule, which is used as an antidiabetic agent. Now the next type of conversions, third compound or third case, already we have seen carbonyl functional group असेल, they are going for phase 1 oxidative conversion. Carbonyl functional group असेल आणि bioreductive enzyme molecules त्याच्यावर act होतील, so it will convert it into the form of the drug molecule which will facilitate the drug elimination. हे झालं secondary conversion किंवा bioreduction, second type of reaction. But if the drug molecule have a presence of nitro and azo compound, so what happen? The reduction of aromatic nitro किंवा azo compound, the reduction होता है generally functional groups and they are lead to the formation of aromatic primary amine metabolites. So if these nitro and azo compounds are converted, so they are converted into its primary and amines derivatives, and aromatic nitro compounds are reduced initially to the nitroso and hydroxylamine derivatives. So what happens if the nitro group is present? See, N double bond O is there. So this type of conversions, they are converted into an nitroso derivative. Further, these nitroso derivatives are converted into an hydroxylamine derivative. So what type of conversions? Reductive reactions, and this hydroxylamine derivative is later on it is converted into an amine derivative. So this reaction is also an bioreduction reaction. So what happened when bioreduction म्हणजे enzymatic reduction reaction in the presence of biological enzymes. So this bioreduction of nitro compound is carried out by who is going to carry out this particular reactions? So this type of is carried out by NADPH, that is nicotinamide adenine diphosphate dependent, that means energy dependent microsomal soluble what type of enzyme? Nitroreductase enzyme, which is present in the liver. That means अशा टाइपचे ड्रग मॉलिक्युल्स की ज्यामध्ये नायट्रो फंक्शनल ग्रुप असेल, नायट्रोसो फंक्शनल ग्रुप असेल, those all drug molecules are metabolized in our body with the help of which type of enzymes? Nitroreductase enzyme. And which organ is involved in the metabolism of such type of liver is the drug organ which is involved in the metabolism of such drug molecules. Now, next one is the reduction of nitro or azo compounds. So, just now we have seen the pathway of metabolism. Earlierly, it is converted into nitro group is converted into nitroso group, nitroso followed by hydroxylamine, and hydroxylamine followed by amine functional group. So they are easily removed out, easily eliminated from our body. And such a type of example of drug molecule is clonazepam. So what happened to the clonazepam? Clonazepam is used to prevent, or it is used for the treatment of epileptic seizures. So this drug is used as an diazepam derivative to treat the epilepsy, that is epileptic seizures. This drug molecules, this nitro group is reduced via bioreductive mechanisms, and this nitro group is going to be converted into its primary amine derivative. So this is the pathway how this nitro group is converted into its amine derivative. So what will be the pathway of bioreduction mechanism? It is converted into its nitroso derivative, followed by hydroxylamine derivative, and followed by amine derivative. So they are converted into a metabolite of clonazepam. What type of metabolite? 7-amino metabolite of clonazepam. So this is with respect to the nitro group. Nitro converted into nitroso, followed by hydroxylamine, and ultimately or finally they are converted into its amine derivatives. This is about nitro. Now how azo compounds are metabolized in our body. So the reduction of these azo compounds will be done by hydrazo intermediate. They are converted into an intermediate molecule. What is that intermediate? Via hydrazo intermediate. What is the structure of hydrazo intermediate? So they are going to form the NH-NH type of linking mechanism, which will subsequently cleaved and reduced to yield its primary amine. ज्या पद्धतीने आपण नायट्रो ग्रुपचं कन्वर्जन बघितलं प्रायमरी अमाइन मध्ये. So what type of conversion will be done to the nitro group? Nitro group is converted into nitroso, followed by hydroxylamine, and finally into a primary amine derivative. Similarly, if the drug molecule contain presence of azo groups, so they are converted into, azo groups are converted into its hydrazo group. Hydrazo means NH-NH type of mechanism or linking will be there, and this hydrazo group is further converted with the reductive mechanism into its primary amine derivatives. Now, such type of drug molecules containing azo type of functional group in its structure. So what is the mechanism or flow of conversion? Azo group converted into hydrazo, hydrazo converted into amine. So the drug molecule which are going to present the presence of azo type of structure is a prontosil. Now what is this prontosil? Prontosil is used as a antibacterial drug from the sulfonamide family. आता antibacterial drug ज्यावेळी सल्फोनामाइड्स अजून डिस्कवर झाले नव्हते, that time this prontosil drug molecule is used as an antibacterial drug of which family? Sulfonamide family. But they are going to give the broad effect as an antimicrobial agent. They are going to affect the growth of gram positive organisms also, as well as gram negative organisms also. That's why in middle of the 20th century, it is used as a better option for the antibacterial or antibacterial agent. And from this prontosil, with the help of some chemical conversions, we have derived a very majority of the class of the compound which is used as an antibacterial agent, that is sulfonamide, which is going to affect the very important chemical constituent from the bacteria, that is PABA. So they are going to affect the conversion of PABA to folic acid. That's why in the bacteria, that's why sulfonamide is used to treat the antibacterial agent. So where we are, so that type of drug molecule, that is prontosil, will have the presence of azo functional group in its structure, and they are metabolized by this mechanism. Which mechanism? Prontosil is converted into its hydrazo derivative, and later on it is converted into its amine derivative, that is sulfonamide. So here they are going to break this particular structure with the help of presence of bioreductive enzyme. Now this presence of bioreductive enzyme will convert this azo compound in its primary amine derivative, that is sulfonamide. Now such type of drug molecules which are going to have the some miscellaneous reduction reaction. आता reduction reaction मध्ये आपण two important main classes we have seen. First we have seen the conversion of carbonyl, that is aldehyde compounds, into its corresponding alcohol. If aldehyde is there, it is converted into primary alcohol. If ketone is there, it is converted into its secondary alcohol. आता just आपण बघितलं की नायट्रो आणि अजो ग्रुपचं रिडक्शन कशा पद्धतीने होतं. नायट्रो ग्रुप्स आर कन्वर्टेड इनटू इट्स प्रायमरी अमाइन व्हाया लिंकिंग मेकॅनिझम किंवा व्हाया कन्वर्जन्स. नायट्रोसो हायड्रोक्सिल अमाइन and then amine. Same azo conversions are done with the help of conversion of azo derivative into hydrazo derivative, and finally into formation of two different aromatic amines. First amine is the sulfonamide and second amine is 1,2,4-triaminobenzene derivative. But some miscellaneous reduction reactions are take place in our body, and those are several minor reductive reactions can also occur in our body. Those are the reduction of sulfur containing moieties, that is disulfide and sulfoxide moieties. So the disulfide functional group containing moieties are metabolized into by a minor reductive pathway. How they are metabolized? Conversion of disulfiram into N,N-dimethyl thiocarbomic acid. So what is the use of this disulfiram? Disulfiram is used to treat the chronic alcoholism. If some person is continuously registered to the alcoholic situation, or he is unhabituated to the alcoholic, so that person we are going to treat with this disulfiram molecule, which is going to cause the unpleasant effect when even small amount of alcohol is consumed. Some uncomfort type of feeling will be there if he is going to consume the alcohol, that because that alcohol is going to react with this disulfiram and convert this disulfiram molecule into the N-dimethyl thiocarbomic acid, which is an unpleasant taste, or this compound is going to give the unpleasant taste. That's why the person is not going to take a further sip of alcohol. Now, some type of hydrolytic reactions, that is hydrolysis of esters and amide. The hydrolysis of major biotransform pathway of drug containing ester functional group. Now, some drug molecules are going to present the ester functional group. For example, for our aspirin molecule is going to have the presence of ester functional group. Now, what happened? This ester functional group is hydrolyzed, and this type of hydrolysis is done in the liver, kidney, and in the intestine. So, ester after hydrolysis, आता ester molecule हा कुणाकुणापासून बनलेला असतो? We have seen ester functional group is formed from the acid and alcohol. आता acid आणि alcohol चं combination होऊन ester functional group झालाय, and if it is hydrolyzed in our body, it is converted into the salicylic acid and acetic acid. So what happened? So the aspirin molecule is converted into its hydrolyzed derivatives, salicylic acid and acetic acid in our body through ester hydrolytic reactions. Now, same type of drug molecule containing ester functional group, that is lidocaine, which is used as an anesthetic to induce anesthesia. So, what happened? This drug molecule is also ester hydrolyzed in our body to produce an elimination product. Same drug example is procainamide, which is used to manage and treat arrhythmia. Arrhythmia means it is used for the treatment of heart arrhythmia. So how it is converted into, and it's how it is metabolized in our body? Procainamide by two enzymatic hydrolytic conversion, slow hydrolysis and rapid hydrolysis. In the case of slow hydrolysis, it is converted into its amine derivative, and rapid are there. So these are the types of reductive conversions. They are taken place in our body for the metabolism of such type of functional group. Now, where we are? So we have seen today that reductive phase 1 reductive conversion. So in that we have discussed how the carbonyl compound containing drug molecules, if they are going for phase 1 reductive mechanisms, they are converted into their corresponding aldehyde derivative, alcohol derivative. If aldehyde functional group is there and it is going for phase 1 conversion, it is converted into its alcohol. The ketone functional group at cell, then it is converted into its secondary alcohol. If nitro group is there, if azo group is there, and just now we have seen if ester functional group is there. So these are the phase 1 hydrolysis and reductive conversions taken place in our body for the metabolism of respective functional group containing drug molecules.